Okay, here we're going to look at enzyme activity. We see in our image here, the amount of energy here required is greater with no catalyst, with no enzyme, and with an enzyme we see that energy is reduced. Even though we're starting with the same reactants and ending with the same products, the amount of energy required is reduced. So we want to look at what can impact our enzyme's activity. So some key parts are the order of amino acids. That's why I showed that picture of the chairs here, showing a very specific order that can impact our enzyme action. The temperature the enzyme is in can help and or hinder our enzymes. pH, certain enzymes prefer different pHs to operate efficiently in. Then we have our substrate concentrations, and then we have our enzyme concentrations. We'll look at these in a little bit more detail here. So starting with the order of amino acids, the wrong order equals the wrong shape equals it not being functional. So you can see here a list of amino acids listed by their three-letter codes here. And the order that these occur in is very important because that's going to determine the shape, how these interact, and that is going to form either a functional or a non-functional protein. So getting everything in the right order is very important so that the proper shape can be had, so that the enzyme can produce the proper function. Temperature. Temperature affects the rates of enzyme activity. There's an optimum temperature. The optimum temperature is the greatest number of collisions between the enzyme and the substrate. For the human body, our body temp's about 37 degrees Celsius. This is typically the optimum temperature of our enzymes. We'll notice here the rate of reaction. As that temperature gets colder, we see that the rate of reaction decreases. What some students forget is that we have our optimum rate, and as it gets warmer, then we notice a large drop-off here. This is because our proteins, or our enzymes in this case, are denaturing. They're falling apart. They're taking on a different shape, and they're not able to perform the proper function. So that optimum temperature is that temperature at which we have the maximum rate of activity. Reduce the temperature, we have less collisions. Increase that temperature, we have denaturing occurring, causing these proteins and enzymes to not have the proper shape. Perfect example of denaturing a protein, causing it to unfold or lose its shape, is in the case of the frying an egg. In this case where we're boiling the egg, there are amino acids and proteins in here. When we boil the egg, we're then changing those shapes, and they don't go back when we cool them. It's another important note to make. Once we change the shape of the enzymes, they don't all of a sudden we cool them and they go back. Once you fry an egg, it's kind of that same change of shape than it was at the cooler temperatures. At lower temperatures, molecules move slower. So there can be fewer collisions between enzymes, and that's why you see here the rate of reaction increases as we increase that temperature. However, there's a certain point here where we're seeing a decrease that's causing a denaturing to occur. Denaturing is that protein's unfolding or losing its shape, and therefore losing its function. We see that indicated right here as temperature increases. We see that increase, that optimum rate's the peak, and then all of a sudden we have this sharp decrease as that temperature goes above that optimum rate. So it's not just a perfect linear curve here. There's that optimum rate, and it slows down due to less collisions, and above that temperature we have denaturing occurring. Now pH can also cause enzymes to denature. Most of the enzymes in the human body function between pH of 6 and 8, but it depends where you're located in the human body. Pepsin, for example, in the stomach has a pH around 3, and that's where it works best. And in the intestines, you have more basic pH around 8. So those enzymes are going to perform better at pH 8. We see that example here. Here, this enzyme has an optimum rate right around 7. You can see as we increase the acidity or decrease the pH, we see that rate of reaction decreases. The same thing as if we increase that pH, make it more basic, we see the same decrease rate here. Now, substrate concentration, so the rate of reaction increases until the point of saturation. So our substrate, remember our substrate in our products, we're increasing our substrate, therefore we're increasing our rate of reaction to a certain point. Here it kind of plateaus or peaks out, kind of will level off at this rate here. We also have the amount of enzyme concentration. So enzyme activity is not always linear. It's based on the amount of substrate. So here we have the time in seconds, the amount of product being produced. We have a burst phase here, and the amount of enzyme located here. So the amount of enzyme can also impact the concentration of the enzyme, can also impact the rate that products are produced. Enzyme shape is imperative. Each enzyme has a very specific substrate that it binds to. As a result, you need many different types of enzymes to be able to survive. There's many different locks, many different doors, you need many different keys. Kind of the same concept here, where all our enzymes have very specific shapes that bind to very specific substrates. Lastly here, to change that shape, we have something called competitive inhibition. The body needs a way to stop enzymes from working at times. So here we have our site, we have our substrate binding to the enzyme, and it's binding just here just fine, and it's able to go through and perform its function. No problem. But we have this inhibition site. So if the body wants to stop this enzyme from working, it can take this molecule. When it binds to this inhibition site, we notice the active site here changes shape. As a result, in this case, the substrate may bind, but the enzyme can't perform its function. So this would be a way to inhibit an enzyme without breaking it down necessarily. This inhibitor molecule binds to the site here and can change the shape of the active site, or can cause this to bind but not be able to be broken. So this is just a way that the normal reaction would occur here just fine, and this is a way that the body can inhibit an enzyme without necessarily changing the temperature, causing denature, or changing the pH that the environment may be in. Because we don't always want enzymes always working and breaking things down per se. This is how the body is able to help maintain homeostasis. It can help regulate certain bodily functions.